So, welcome to the third part of the first week series of lectures here in Next Generation Sequencing Analysis. Hopefully you've had a chance to look at the other couple of lectures coming into this one. In the last one we talked a little bit about the different applications of next generation sequence data being applied to. But today what I'd like to do is to really talk to you about the meat of the course and what this course is really all about. What this course is about is taking you from the sort of the raw DNA sequences that a biologist would collect in a lab, and then eventually taking it here to a final, nicely completed and analyzed whole genome sequencing data set. And really, your job as a bioinformatician and the person doing the next generation sequence analysis is in this phase of the project right here. And what I'm going to introduce you today is some of the basics of what does this step actually look like. Before I get into that, however, I do want to give you some of the background of this concept of shotgun-style sequencing and its basic rationale. This is what we call a pileup. This is again an integrated genomics viewer screenshot. I think I mentioned pileups in the previous lecture. But once again, these are the reads that have been aligned to the reference genome. And in a particular position here, you can see a SNP, heterozygous SNP. But really, the concept here behind next generation sequencing is the idea of coverage depth. So let's talk about that just for a minute, and I'll give you a rigorous definition here. So coverage is the average number of times each base is sequenced in a genome using this shotgun style of next generation sequencing. And what I want you to think about coverage as is really just imagine, don't get into the algebra, let's just talk about it for a second. Coverage depth is really an experiment. If you just imagine stringing together all of the short reads from an entire sequencing run, okay, and summing up the entire length of bases that were sequenced. And then imagine dividing that number by the size of the reference genome, and that is effectively the coverage depth. Okay, so in math terms, coverage here, again, this is the average depth of reads at each position in the genome. You take the total number of reads and you multiply it by the length of the reads and divide by G, the genome size, which again is typically the size of the reference genome or a haploid complement of the genome. So I remember being a little bit confused by this several years ago myself, but coverage is sometimes used as a strictly theoretical measure. So this coverage is something you can refer to before you even begin the sequencing of a genome. So if you know how many reads you anticipate to get off of a sequencer, say from a lane of high-seq sequencing, you can talk about the coverage depth that you anticipate in the sequencing experiment. Right? You simply take the number of reads that you expect, multiplied by the genome size, multiplied by the length of those reads, and you know your reference genome size, so you can calculate the coverage depth. However, more typically and maybe a little bit more useful is to talk about the aligned coverage depth. Okay, so after you've mapped the reads to the reference genome, inevitably these numbers change from your theoretical expectation because much of the genome is not mappable. Okay, it's repetitive, for example, is one reason why the genome is refractory to mapping. And so it's and you just lose a lot of reads for various reasons, maybe low quality reads, etc. So here, for the aligned coverage depth, the formula is identical. We simply just replace now here the number of aligned reads for n rather than the total number of reads. So imagine we have 300 million aligned reads, each of length 100 base pairs. That gives us 30 gigabases of total sequence, and therefore 30 gigabases divided by 3, the human reference genome size, gives a coverage of 10x. Okay, so I really I'm missing a slide here, which is very strange. Let me just, if you will, just hang on for 1 second, because that is the slide which, very strangely, my computer skipped over, but there it is nonetheless. So I wanted to give you a sense of some of the rationale for the shotgun style of sequencing. I'm not going to ask you to know any math in this class, obviously. It's not really the nature of the subject. However, this really is the basis for the shotgun style of sequencing. So the question is how do you know how much of a genome is expected to be sequenced giving a certain coverage depth? So what proportion of the genome will not be sequenced if you say sequence at 10x coverage depth? Well, that question can really be answered using the Poisson distribution. And Lander and Waterman were the first ones to recognize this. So Py here, P as a function of y is the probability of a given number of events, or probability of events in a space or time interval, and lambda is the mean number of events per interval. In next generation sequencing analysis, P sub y is the probability that a site will have a certain number of reads or a certain depth at that site, and lambda is the mean depth of coverage here. And y is the coverage depth that we're interested in calculating, the proportion of the genome that has that coverage depth. So given the coverage, then, we can calculate the probability that a particular genomic position will be covered at a certain, by a certain number of reads. So that turns out to be a really useful thing. So here, just an example, let's imagine that we have 10x coverage, that we've sequenced our genome. So lambda here equals 10. And we want to know what is the proportion, say, of sites that will be covered at exactly 3x. So if we look at this equation here, again, going back 1 slide, okay, P sub y, so y here is 3. That's the desired level of coverage depth. And lambda here, okay, in this is 10x, or 10. Okay, so we substitute 3 and 10 in this equation, and this is factorial, so this is 3 times 2 times 1. Here you can see that here in this equation here, or as I substituted in here. And so the answer to the question is at a coverage depth of 10x, what proportion of sites will be covered at exactly 3x is 0.00757. Okay, interesting. So the real question then might be what proportion of the genome won't be sequenced at all. So when we're doing a sequencing experiment, we really want to know, okay, if we collect X amount of reads, say enough for 10x sequencing depth, coverage depth, what percentage of the genome would we expect to not be sequenced at all at 10x coverage? Well, the answer is a very small percentage. Okay, so less than 1 in 10,000 sites in the genome we would expect to not be sequenced. So again here, just notice that instead of substituting 3 for y here, what I've done is to substitute 0. Okay, so here's 0. The 0 factorial, by the way, is 1. And so here's the 1 here. And so you can do the math on that. Just plug and chug, really, and the answer is less than 1 in 10,000 sites would not be sequenced at 10x. Okay, so what proportion of the genome then is expected not to be sequenced? So this is the proportion. So how many bases then would be expected to not be sequenced? We simply then multiply that proportion times the genome size. Okay, and that would give us the answer of about 135,000 base pairs would not expected to be sequenced. So this is all theory, right? But this gives the theory for shotgun sequencing. This is why shotgun sequencing works, is because by random chance, even at a fairly moderately low coverage depth of 10x, we still expect the vast majority of the genome to be sequenced. Of course, that doesn't mean that the vast majority of the genome can be sequenced. Okay, this is theory. Reality is there's repetitive sequence in the genome. There's insertions and deletions in the reference relative to the sample. There's a lot of things that make it so that reads don't map to the genome. So in fact, after you align your reads, you find that there are huge chunks of the genome that are not sequenceable. So this is just the theoretical basis. We expect everything to be sequenced. It doesn't mean in practice that everything is in fact sequenced. So I'll leave you to do the math here. What proportion of sites will be covered at 3x or less? As you might have guessed, you simply have to fill in this equation then for 3, 2, 1, and 0, and then sum, okay, the proportion in order to answer that question. Okay, so I've just quickly done the equation here for you for different levels of coverage depth. Okay, what proportion of the genome would we expect to not be sequenced when y is 0? Okay, so when you only sequence at 1x coverage depth, notice that percentage of the genome that's not covered at all by any reads is 37%, or 0.37 fraction. Okay, and of course this is just 100% minus this value here is the percent that is sequenced. Okay, so at 10x, obviously, the vast majority of the genome is sequenced. So let's switch topics to another very important basic idea, and that is the idea of base quality encoding in the raw data that comes off of a sequencing machine. This goes back to the old days of what we called Sanger-style sequencing. Some of you may be aware of Sanger-style sequencing and how it works, but effectively, in any kind of modern sequencing application, we need to incorporate error probabilities into our predictions of what a real base is and what the likelihood is that we will make a mistake in calling a base a particular nucleotide and actually being wrong about it. And Fred quality scores are a scaled error probability that are quite easy to understand and quite intuitive. So the equations, again, you might find to be a little bit daunting perhaps if you're not into math, but I think you'll understand here in a second that this is actually pretty intuitive. So if you know the error probability and want to know the Fred scaled quality, you simply take p, this is the error probability, you multiply it times minus 10, the log of that error probability, and that gives you the Q, or the Fred scaled quality. Okay, this is simply the reverse. If you know the Fred quality, you can calculate the error probability. Okay, and using this equation here. So this is really simple. Fred quality 10 means that there's a 0.1 probability that the base call is not real. That's 10%. Okay, probability that it's an error. Okay, so Fred quality of 10 is not a good Fred quality. A Fred quality of 20 is one order of magnitude, okay, lower probability that this is an error. Okay, so in other words, a 20 is better because it's a lower probability that it's an error. Okay, so 30 is 1 in 1,000 probability that the base is an error, and 40 is 1 in 10,000. Okay, probability that it's an error. So typically in a sequencing study, our qualities are 20, 30, or somewhere in this range, sometimes higher, and frequently cutoffs of like 20 Fred quality are assumed to be fairly decent, and that would give us some false positive rate at the raw nucleotide sequencing level of about 1%. Okay, so you do see these Fred qualities reported frequently. So just having, you should be able to immediately remember that if you see a Fred quality score of 20, that means it's a 1% chance that there's an error, and you can calculate that, okay, by simply taking and replacing Q here with 20, and that will give you the error probability of 0.01. Okay, you might want to convince yourself of that, with a calculator. So, I should point out here some basic properties of sequencing experiments. This comes from really my own experience, and I think it'll be really useful for you to sort of familiarize yourself with some of the basic things that I worry about, and anybody handling raw sequencing data should be aware of. Okay, first of all, error rates can be high. Okay, so there are a number of reasons for this. One has to do with something that's totally out of the control of the bioinformatician, and that is errors that get incorporated during the library preparation step. Okay, so these are problems that are not well understood exactly the biochemistry as why, but the DNA can become altered during the library preparation step, and then during the PCR step. PCR is not a perfect technology, and nucleotides get misincorporated by the Taq polymerase, and so these are things that happen upstream of the actual sequencing process itself, and so errors get incorporated early on without, it's not anybody's fault in doing any analysis or not taking into account the base qualities. The base qualities are actually the qualities associated with the sequencing itself, and we generally ignore the problems that are associated upstream of the sequencing run. I'm not going to, that's something that is fairly inside baseball, and I won't be talking about these types of things too much throughout the class because there's really nothing we can do about it. Just understand that there are different phenomena that cause high error rates in next-generation sequencing data. Another thing to be aware of is that there's frequently a GC bias in the sequencing of short reads, and the reason for this is due to biases, again, in the polymerase chain reaction, or PCR. So PCR doesn't work well in really high GC content regions of the genome. So I'm working on an organism right now that's one of the highest GC content organisms out there, claiming to own as Reinhardt. Has a genome wide GC content of 65%, and the really high GC regions above 70 actually really don't sequence well at all. And that's simply because the Taq polymerase doesn't amplify them, so those regions never get represented on the flow cell and never get sequenced. So there is some GC bias in our sequencing. Another thing to keep in mind is all of the base quality, Fred quality scores that I just sort of described, how we calculate them. It's important to realize that they're not calibrated, okay, when they come out of the box or off the machine. So what that means is, while in principle, if we go back a slide here, in principle, okay, the error probability is related to the quality score. This math is, in fact, exactly correct. The problem is that this error probability is not well known. Okay, algorithms predict what that error probability is, but they can be wrong. In particularly, uncalibrated means that those error probabilities are two error probabilities may be tenfold different in magnitude. The problem is we don't know exactly what any one error probability is. Okay, so relative to each other, two error probabilities might be an order of magnitude off. The problem is that saying that one base has an error probability of 0.1 is very difficult to do without some post-sequencing calibration. All right, I can answer any questions you have about this. I certainly won't test you on this kind of thing. It's just something that in the human SNP calling literature, you'll find a lot of reference to calibrating the base qualities. So what that means is, again, just determining empirically, rather than relying on an algorithm that's just predicting. You have to determine empirically what those error probabilities are. Okay. Base qualities, however, even though uncalibrated, are very useful because we think that most of the time they're probably close to being correct. But one thing that we noticed very clearly in virtually all, at least Illumina runs, is a pattern like this. So here we have the Fred scaled base quality. So a 40 means a 1 in 10,000 probability of an error. Whereas, so here's the green zone. This is a FastQC output. We might get a chance to run some FastQC. I haven't decided yet if that's something I want you guys to do, but it's very easy to do. So green here is the green zone. That means this is good sequence, right? Above between 30 and 40 Fred quality scores. 40, 1 in 10,000 probability of an error. 30, 1 in 1,000 probability of an error. So what you're looking at here is cycle 1, cycle 2, cycle 3, all the way to, these are 150 base pair reads, which is 150 cycles of sequencing on the flow cell. So remember, on the flow cell, an image gets taken, okay, at after the first cycle, and another picture gets taken after the second cycle. And what we're seeing here is the a full single end 150 base pair run. So why do you get this drop in base qualities? Well, each cycle there's some probability that the polymerase that's extending the nascent molecule, at the end of which is a terminator that is fluorescing and is being captured by the camera, there's some probability of an error, okay? Or say that 2 nucleotides get added when only 1 nucleotide should get added to the growing molecule. What happens is, as a result of that, is that no base can get incorporated in certain at a particular cycle in a particular cluster. So what happens is that over those error probabilities of misincorporating or not incorporating a base start to add up, and the later cycles then the clusters give ambiguous signals as to what base is at that particular position, because different reads within, or different physical molecules within that cluster actually have a different color. And so that's noise, and so the machine can't distinguish between the different colors associated with the different terminators. So anyway, just be aware, probably a little more detail than you need to know, but be aware of this pattern. Okay, some people decide they want to trim the reads before they align. I generally don't, and there's now empirical evidence that can actually increase the error rate, because the more you trim, the less likely that the read's going to actually map to the correct position in the genome. And so what I generally do is map the reads to the genome, and then let the SNP caller or the other algorithm that we use on next generation sequencing data take this error into account, and we worry about it later. Okay, so that's what this here is. So I hope this is clear. This is where one case where online lecturing is a little bit more difficult than if I was sitting there with you in the classroom, because you could immediately ask me questions. But so these are things that anything you come down, just jot down on your notepad, questions you might have, and I can certainly try to answer them for you in our upcoming webinar. So this was one of the questions that I thought to include here in this lecture because it was something that I asked and desperately wanted to know when I first started learning about next generation sequence data analysis. Like, what do I see? Like, as the informatician, as the person handling the raw data, what do I get when it comes off of the machine? What does it look like? So we'll talk a little bit over the next few minutes about formats, and we'll talk really we'll be dealing with these for the whole class. So we'll, repeatedly come back to these same formats, and we'll get to know each time we come back to them, we'll get to know a little bit more about the formats. But here's kind of an overview. I'll give you some detail here. So the answer to the question is, what do you get? Well, you get the FASTQ file. Okay, I'll tell you exactly what that is in a second. We're frequently dealing with SAM or BAM alignment files. And then finally, when we get to the end of our analysis, we end up with variant call format, or VCF, which is how we store all of our SNPs and structural variants. So this is what the very raw output that typically most informaticians see after a sequencing run. So recognize there is an upstream step. So all of these images that come off of the sequencer all have to be processed. Okay, the multiplexed or barcoded reads. So if you've say sampled 5 different samples of RNA-seq data in a single lane, those are all intermixed on the same flow cell. So one cluster can be from one cancer cell, another cluster right next to it can be from a different individual, say from the same hospital that happened to be on that same sequencing run. But those reads have to get computationally parsed out by someone, and that happens upstream typically of the FASTQ files. So what happens is Illumina has software called Cassava. You'll hear of that someday when you work with Illumina data. Well, what Cassava does is it takes care of all that for you. So it parses the data so that each sample, okay, then gets delivered to the informatician as a pair of FASTQs. So in a paired end run, sample 1, even from a multiplexed run, would appear as 2 FASTQ files, a forward and a reverse for the paired end. And sample 2 would then show up in the informatician's directory as forward and reverse for sample 2. Okay, and et cetera, et cetera. So usually you already have everything sort of parsed here for you. So a single FASTQ file and its paired file come from a single sample. So what's FASTQ look like? Well, this is the basic of it. You have a sequence identifier. You have the DNA sequence of this is a short read, probably 50. I don't know. I didn't count. Historically, there was an additional line here that had an additional identifier, but now we just have plus here, and then we have the base qualities. Okay, so these are symbols that represent the Fred quality scores that I talked to you a moment ago about. So remember that towards the end of the reads is where the error probabilities go up, and the Fred quality scores go down. So if you really want to know the details, these are characters are ASCII encoded 33 plus the Fred quality. I don't anticipate asking you detailed questions about this. However, I would like you to take a look at the Wikipedia page. This is the actual place where most people refer anyone to when they're interested in the FASTQ format. Just realize these are what we call Sanger. We call these Sanger qualities. Historically, FASTQ was kind of a mess, even up till about 3 years ago. There was several different FASTQ formats, but hopefully that's all in the past now. Unless you're dealing with a very old data set, these symbols didn't actually mean the same thing across different FASTQ files. As long as you have Sanger FASTQs, what we call Sanger FASTQs, then you're good. Okay, so but have a look at the Wikipedia page if you want to understand more about about the FASTQ encoding. So this is what we call the four-line style of FASTQ. There is what's called a multi-line that occasionally you run into. Typically not maybe raw reads, but maybe if the reads get longer, we'll have to watch out for this. What multi-line FASTQs are is when the nucleotide sequence extends onto a second line of the file, and the base qualities extend onto a second line also, and that becomes difficult to parse computationally if you're trying to write code to do it, and certain software may not even recognize it. So I'll show you how to quickly take a peek at some of the files, and I'll give you some FASTQs to take a look at, and we'll get to play around with some of this. So, but always it's a good idea to look at the raw data before you start using it. So FASTQs, we align those raw reads, that's what the raw reads look like, to a reference genome, and we end up with a BAM or SAM formatted alignment file. So SAM or BAM here is, so the SAM is a raw text, BAM is a binary form of an alignment, and, excuse me, sorry about that, my computer keeps racing forward. So in a BAM file, this is a tab delimited. Let's talk about SAM, because BAM you never really look at. You always have to convert BAM to SAM and actually to visualize it, because BAM is binary encoded. So SAM is the raw text, which we're looking at here. So it's a columns of data. So on the leftmost column, column 1 is the read name. Okay, that typically is coming directly from this sequence identifier here in the FASTQ file. So here we've aligned it to the reference genome, and the purpose of the BAM is to store information about how that read aligns to the genome. So this is where it's aligning. This is, say, chromosome 1. Notice here that they're actually skipping a couple of pieces of information here, which we'll come back to in the class, and I'll actually give you an exercise on how to interpret the BAM file, because it's really important skill to learn how to manipulate a BAM and to understand exactly the content of information that's in it. So this is the alignment itself. So there's 23 matching bases, and consecutively, I actually don't know what the N here is. It could be 6 bases that couldn't be determined, and then 10 more matches. Here's the read sequence itself. So this is the A's, T's, and G's and C's, from the FASTQ data. And then here's the base quality encoding, the base quality encoded data here. And then there's this metadata, or data that refers to the read. So this is oftentimes a liner specific, but there's these tags, okay, that give information, about whether the read was, say, uniquely mapped, or whether what type of sequencing machine sequenced that read, etc., etc. Okay, so I thought I'd alert you to the fact that BAM is a quite sophisticated format. Here is what we call the BAM header. Okay, so this was BAM, for an individual read. This is the BAM header, and the BAM header stores an enormous amount of information that's important for downstream software to correctly do the SNP calling or variant calling of different types. So at the top here, this is an important tag here. This indicates that the BAM is coordinate sorted. Okay, so BAM can be sorted by coordinate, or it might not be. So if it's coordinate sorted, you have chromosome 1 first, and then you have reads that map to position 1, all lined up together, then reads that map to position 2, all lined up together. 3, 4, 5, etc. in consecutive coordinate sorted order. But sometimes software is developed that doesn't use coordinate sorted. It rather needs read name sorted. So if you coordinate sort a BAM, realize that the reads themselves for a paired and run are actually the two reads that make up the pair are now dispersed, okay, across the BAM file. Okay, so they're not next to each other anymore in the BAM, and that can cause a problem if the algorithm needs to identify the two pairs. Okay, so these files are enormous, and this is an important thing to understand, that these files can be 100 gigabytes on your computer. Okay, they can totally take up your entire hard drive if you have a large sequencing experiment. So some of my BAMs, they're merged BAMs where all of my samples are in the same BAM, 20 genomes, and that BAM file is more than 100 gigabytes. So no computer can store all of the information about those reads in a BAM file in memory. Okay, so a computer, without being able to store them in memory, essentially cannot know whether two reads from the same paired N fragment are actually in the BAM or where they are. And so in order to deal with that, sometimes people actually sort then on the read names so that the BAMs then contains reads, paired and reads that are physically next to each other in the BAM. Okay, so I could kind of get lost here in detail. So I think I'll try to just alert you to the fact that you have different symbols here, or different tags that have different pieces of information. So these SQ tags give information about the reference genome itself. Okay, so here are the different chromosomes in the human genome. Chromosome M, that's mitochondria. Chromosome 1 to 9, 10, 11, etc. So it tells the software that's reading this BAM file what to expect in terms of the different reference chromosomes. Here, the read groups. This is important for handling information about a particular strategy of sequencing. So sometimes in human genomes, when you do whole genome sequencing, they're so large that in order to get deep coverage, you have to sequence across lanes. Well, FASTQ files come, in a single lane, from a single lane, and also you have to correct for certain types of biases on a lane-by-lane basis. Okay, so we'll talk about duplicate reads later, but just take it for now that there are certain things that have to be manipulated on a lane-by-lane basis. So if you have a sample that was sequenced across lanes, there are some pre-processing steps that have to take place on a single lane of data. You can't just merge the FASTQs from many different lanes for the same sample and then proceed as if they didn't come from different lanes, because certain corrections have to take place on a lane-by-lane basis. So what happens is this read group, this ends up becoming a tag here. So this is a single reads entry or record in the BAM, and here the read group is one of these tags that I was referring to in the previous slide, okay, or somewhere back here. These metadata tags, okay, contain the read group. So the read group here is 20FUK.1. This is 20FUK.1. And notice up here is all this information about 20FUK.1, like it was Illumina. This is the library. I don't know what PU is. Some of these can be quite rich in information, but this little tag right here tells you all kinds of information about how that particular read was sequenced and where it came from. Okay, so hopefully that's just an introduction here to how these read groups work, but they give all kinds of information about an individual read. Finally, these PG tags give some sense of
what we call data provenance. Okay, this is what has been done to this file. Okay, so this says that BWA was used, the Burroughs-Wheeler aligner was used to do the alignment, and then GATK was used to do the recalibration of the base qualities, presumably in the input FASTQs. Okay. So we'll come back to that. I mean, this is, again, this is a time where somebody might have, you might want to raise your hand and ask a question. So write down your questions. We'll talk about it. Probably the best thing to do is to just get your hands dirty here on some BAM files, and I'll be describing in the homework section about how you can download some FASTQs and begin the corresponding BAMs, and we'll be giving you an exercise where you can begin to use what's called SAM tools to dissect apart a BAM, and you guys are going to become BAM file ninjas, and you guys are going to be able to shred a BAM with no problem at all once this course is completed. All right, so the variant call format, this is the one that I spend the most time working with. So after you spend some time processing your BAMs, getting comfortable with there's fairly standard procedures, right, to how to process a BAM, and eventually you end up spending, I think, a more significant amount of time dicing of VCFs and understanding the information that's in a VCF formatted file. It's a very rich format, storing variant information from many sample genomes together. It can handle both SNPs and insertions and deletions and translocations and other types of structural events. So it's a very rich format, to say the least. So I'd like for you to check out this website, okay, guys? This is something where over the course you'll want to really digest what variant call format is, as you will BAM format, and the 1000 Genomes Project website is the place that hosts the VCF format, because it was really the format was really constructed basically by the people behind the 1000 Genomes Project, and now everybody uses it. Okay, this is the standard. Both of these two formats, well, all three now, FASTQs, BAM, and VCF, are the standard. There really is nobody who uses any other format for handling the raw data, the FASTQs, the aligned data, the BAMs, and the polymorphism data in the VCFs. I mean, there are other formats for handling VCFs. I mean, a simple table of SNPs is one way to do it. And yet, for next-generation sequencing data, massive amounts of data where you need to keep track of all kinds of quality information, the VCF format is certainly the format of choice. So, once again, we have a header. I'll, I guess, sort of explain some of this now, but a lot of this is just introduction, and we'll come back to this later. This tells the file format, version number, the reference genome that was used to align the reads originally. Then there's these info data lines here, which are referring to the info column. Okay, this thing is wrapped here, so a little disturbed that my slide here is wrapped, but this is one line. Okay, take my word for it. Info, then format, then NA401, NA40s, then 2, NA40s, and 3. These are the sample genomes here. Okay, 3 human genomes. So column 1 is chromosome, column 2 is position. This is the physical position of the nucleotide. Column 3 is the the SNP identifier here. This is it here. RS6054257 is actually a name assigned to a particular SNP. Okay, so chromosome 20, position 14,370, the name of the SNP. Here's the reference base. That's what's in the reference genome. The alternate base of this position is an A. Here's the quality. This is a Fred-scaled quality that there is a variant at this position. So there's a 1 in 10,000 that's not right. A 30 is 1 in 1,000 roughly. So 29 is roughly 1 in 1,000 probability that this is actually an error. This pass here is referring to the filter, so this is quality here. The filter here is pass. I'll describe that a little bit later, but a read failing of some filter would not have pass here. It would say, have a qual 20, say Q20 might be a filter, and that if this was not a 20 or above, it would have Q20 here. That's something that the user can impose, yourself. Here's the depth of coverage, okay, across these 3 samples. So if we sum the number of reads across samples, okay, we end up with 14. Over here, we get the depth for each individual sample. I hope those numbers add up. Yes, they do. We'll get to that in a second. Here's the allele frequency. It's at 0.5 in the population of individuals, so 3 individuals. We'll come back to that. So here, let's look again. Maybe I'm going a little too fast, I don't know. Here's the quality, that's 29. The filters pass. The info data are here. And notice we can relate the info to back here. So info here is depth, AF, and dB. We come down here, we have depth, AF, and dB. But here we get some explanation for what those values are. Okay, so here total depth is what dP refers to. Okay, dP is total depth, and AF is the allele frequency. dB is dB SNP membership. In other words, has that SNP been discovered before in some other data set? And I think that's just a switch, meaning it doesn't have a value. Okay, so here these numbers here tell you what type of information it is. I actually don't know. I don't remember what the 1 and the A mean here, but take my word for it. These have valuable information, so that if somebody was writing, a software engineer was, needing to distinguish between a decimal value, say, and an integer value, that would be recorded up here. And this is just a switch, and presumably that's recorded up here too, meaning that it doesn't have a value associated with it. It's either there or it's not. Notice down here, there's no dB here, right, or here. All right, so anyway, importantly is the format. So the format column here, this tells you what values are in each samples column. So VCF is 9 columns of data, okay, plus however many sample genomes are in your data set. This one has 3, so it's 1, 2, 3, 4, 5, 6, 7, 8, 9 columns, followed by the 3 sample genomes. So the format field here is GT, GQ, and DP. So that's telling us that there's 3 subfields in the sample genomes. Okay, so subfield 1 is GT, which if we look up here, what is GT in the format field? Okay, GT is genotype. Okay, what is GQ? Genotype quality. That's a Fred-scaled quality, again, of a genotype being an error. Here's the depth, the read depth. Okay, so let's look at that. So here's GT, GQ, DP are the 3 subfields. Here in sample 1, this is corresponding to NA00001 genome, 0/0. What this means is 0 is referring to the reference base, okay, G. So this guy is a GG homozygote. The quality of that genotype is 48. Okay, so that's the error probability is less than 1 in 10,000, and 40 would be 1 in 10,000. This is a lower error probability, higher Fred score, meaning lower error probability. And the depth is only 1 read. Okay, so here this individual is a heterozygote for 1, 0. So 1 here is an A, 0 is a G. There is a genotype quality of 48, and there are 8 reads here. This individual is 1, 1, or alternate, okay, alternate, so here's alternate, this is reference. this is a alternate allele homozygote, so two alternate alleles. The GQ here, the genotype quality, is 43 and 5 reads as the depth. So remember, over here we had a total depth for all samples in this one VCF record at this position was 14. So let's see if this makes sense. So here we have DP here. This is 1 plus 8 is 9 plus 5 is 14. There's the 14. Okay, so you can check this out some more, but for now that's a fairly detailed entry to VCF format. VCF will get into in several weeks from now, where we'll begin to carve up these, and you can become VCF ninjas just like you'll be BAM file ninjas. But anyway, for now, that's what I wanted to share with you. Hopefully that gives you some insight into some of the details that I know you guys are just chomping at the bit to get at. So that's it for this third lecture, and I'll see you guys in the webinar. Okay, see you soon.